Good afternoon. So today's my talk is titled Adventure. So I'll bring you across adventure across skills. But before I do that, I want to give a brief introduction of my background and where we come from and why we think this is an adventure. As you have heard, I'm a chemical engineer by training. And as a chemical engineer, we were really interested in seeing how we could use microbes as microbial factories, whether it's for health or sustainability. So in order to do this, we actually look towards the central dogma. So this is this figure here is a extended picture of the central dogma. You have the DNA going to RNA and then going to proteins. But proteins don't just stay as proteins. Proteins actually make small molecule. They are biocatalysts that can do chemistry. They make small molecule which then affect the cell, which also affects DNA, RNA. And again, regulation within the cell. So in this talk, we'll talk about how we are trying to engineer two things: genes to molecules and also genes to proteins. Much of this talk will be focused on genes to molecules, but we also have a short story on genes to proteins. So question, what the molecules that we are looking at are actually secondary metabolites. They're not primary ones. They're not required for life. Rather, they are required for chemical warfare. And if you can look at the structure of the molecules, they are very complex. They are 3D structures, they have complex chemistry, and they are highly bioactive. So you will see examples such as erythromycin or and also dopamycin, and a few that you can actually recognize in the drugstore. And about, I would say, of drugs, about 50% are actually natural product or natural product derived. So what's so interesting about this natural product? So although they are very complex looking structures, you can always link the genes back to the chemistry. So in this case, this is the chemistry, or these are the genes involved in making natural product. What you can see is that each gene is in charge of different modular, and it's like a factory assembly line. So you know which part makes the wheel, which parts make the steering wheel, the seats, and then you can modify accordingly. Or that was the dream, right? Is to modify the genes, then which will then affect the molecules. If you could affect or place the Lego in the correct conformation, you can get a designer molecule, for example. And then a lot of these gene classes actually are found in actinobacterians, fungal, etc. But in this talk, we'll focus on the actinobacterial. So because I say this is an adventure, and every adventure there's going to be a problem, okay? So the first problem is, so in theory, we are able to swap genes to make new molecules, okay? But this is sort of like the meme of what we think is happening. So you would think that I can do, from any gene, I should be able to make the corresponding molecule, right? But this is what happens in the lab. With every gene, there are problems such as the genes are repressed in the cell, or there's not enough substrates. And then you will get, maybe you only see a small subset of molecules being expressed or produced. But here's what we really want. We want to be able to super energize the cell to make all the possible molecules it can. So basically, be able to sample the full diversity that nature has, right? And so this is the quest, right? In the venture, we want to be able to sample nature's full potential. And how do we go about doing this? This is what can happen. So in a native strain, like I mentioned, there are limitations based on culture, fermentation. There are also limitations based on gene regulation. And so there are various strategies to upregulate either within the cell or on the biosynthetic cluster itself. And here, really, the first part of our quest, we will look at gene activation. Gene activation is nice because there's permanent upregulation, but it's also nice because it would target the gene clusters of interest. So one gene cluster of interest that we are very interested in is the polycyclic tetramate macrolactams, so PTMs for short. These are very short or 20 kilobase pairs gene cluster, and they consist of multiple genes. The longer one would be a hybrid PKS and RPS gene. And they really make various diverse compounds, as you can see over here. And this would include compounds that are antifungal, antibacterial, or even anti-cancer in nature. So in nature, from genome mining, you should be able to find these clusters. So what do you do when you find a cluster of interest? What people have done, I would say in the early 2010s, would be, or even now, we'll take the gene clusters out and we'll refactor them, right? Because we refactor them for a different host strain. In this case, this was done for yeast. It was refactor. There was different combinations of refactoring, and then there was the expression in yeast. And then what happened is that, then after that, you get the compound, and then you isolate and you characterize. But how can we do this better? And like, what tool can we use to make this better? So this is where in early 2010s, there's CRISPR-Cas. And for CRISPR, maybe a short introduction of what CRISPR does. You need a CRISPR protein. You need a guide RNA. You need a repair template. The CRISPR will introduce a double-stranded break at a specific site on the genome. With the repair template, there will be a homologous directed repair where you can then insert your fragment of choice into the genome. So this could be a deletion, it could be an addition, and of different lengths. So with CRISPR, what we did with, and this is with Hui Minzhao over at UIUC, and also with Mingzi Zhang, we did, starting from his technology, looking at CRISPR for streptomyces, we then turn it into an activation tool, right? Where basically now, instead of doing the multiple steps, as you can see on the left-hand side, all you need is a single insertion, and this is a 100 base pair insertion promoter in front of the open reading frame to upregulate all the genes in the gene cluster. And basically with one edit, you are able to see new upregulations of product being formed. And then with really this is new PTMs really characterized with MS and MMR after that. So with the CRISPR-2, what was very interesting for us is that actually this was using the I think the most common SP-Cas9. But we also wanted to say, can we look at other Cas proteins for streptomyces? And over the next few years, we did Cas12 or CFP1 protein, and also Cas9 proteins of different sizes and also of different PEM specificity. And basically now we have a toolbox of different Cas that we can use for engineering, and we have tried this across Actinobacteria. But what is so interesting, and why do we need a toolbox, right? Because Cas9 seems to work fine. But the reality is that Cas9 does not work for all strains. And this is, some data that we have that Cas9 itself is toxic. It actually doesn't transform very well into strains. It actually kills strain, and possibly because of off-target of double-stranded breaks. But also depending on the context of what you're trying to edit or your strain, different Cases actually have different efficiency. So it's just why we needed a Cas toolbox. And we do have a Cas toolbox right now. So in all, I think we have established our Cas-mediated protocol. So what can we do, right? So now we have our tool in hand, and we need to continue our quest, right? So what can we do with this toolbox? We can do engineering, and we can do discovery. So the next few parts will be really our applications on basically using the Cas, right? The first application, we wanted to look at, so we were screening in our own library for antifungal agents. This one molecule, which is a 32-membered ring, 32-membered glycosylated macrolactam called nitrosomycin in our own strain. So this is an in-house Singapore-derived strain. We label it A793. We could see the appearance of nitrosomycin. So this is a compound that was known previously, but really there was no genomic data to support how it was being made. So with this strain, we did PacBio sequencing and managed to decipher how the strain was being made, what the assembly line looks like. And what is more interesting, that in this strain, we also saw a new analog, because this we labeled nitrosomycin B. And what nitrosomycin B and A have difference is the sulfonation over here. But what's interesting with this nitrosomycin B, we also have higher activity. So there's higher bioactivity. So that was very interesting for us. But then one question was, how can we make more of this nitrosomycin B or even nitrosomycin A? So to do that, we actually turned to CRISPR. We use CRISPR to knock in constitutive strong promoter in front of the gene. This is activator that's specific for nitrosomycin BCG. And so with this knock in, and you can see this is, so on the left-hand side, this would be the qPCR, the gene expression levels after knock-in media, SV2 media. And then on the left-hand side is actually the abundance of the product. A is in blue and red is B, between mutants and wild type. So we see fold change. This is increased threefold. But notably in a media that doesn't even make nitrosomycin, so let's say CA10LB, right? In this case, we see upregulation again. But if you look at wild type versus the mutant, basically being able to activate an activator, we now have 12-fold improvement in yield. So that was very significant. And again, this is incorporating a 100 base pair sequence into the genome. And if you remember, we have the A and the B. The B is supposed to be better. So in order to get more of the B and really none of the A, what we then did was then we took the sulfur transferase, so we could figure out what the biosynthetic pathway was. We took this gene and then we inactivated it, or we deleted it. Then with this deletion, what's actually very interesting is that we not only see that we have no more A formation, A analog formation, but we see that the B analog formation actually is significant now. It's also, so we expect maybe keeping the same amount of B, right? Or even maybe the amount of B plus A, but actually we got much more about the upper that of what we expected. And that was a very nice event for us. But really there is a lot of, again, this tells us there's a lot of moving parts in the cell, that perturbing one doesn't, always give you the expected outcome, but this is a very nice expected outcome for us. So we continue on this strain, and what we saw in this strain when we actually knocked down nitrosomycin. So remember, nitrosomycin was observed in this strain initially. We had to do deletion studies to make sure that we know the corresponding gene cluster for nitrosomycin. So in this case, when we knock down, so this is the, and where my arrow is, this is the wild type nitrosomycin profile. When we knock down the strain nitromycin, what we see is this new peaks appearing. And these new peaks are actually amisporol. There are very potent polypeptide antibiotics against gram-positive and also multi-resistant gram-negative bacteria. So this is a very interesting molecule by itself based on this bioactivity. And simply by shifting the ratios of what the strain can make, it actually wants to make something, right? So at this point, by knocking down one, you actually get upregulation of another compound. And to further upregulate this compound, we actually look at different strategies. Again, we try to upregulate genes in the biosynthetic gene clusters, but what turned out to be the most successful was really upregulating fatty acid CoA synthase. This actually regulates primary metabolism, and what it does is that it increases the CoA pool of substrates for amisporol. In this case, we have, compared to wild type where we had almost nothing, we now have upregulation at 97-fold. So with CRISPR and basically being able to tune, inactivate, delete, we were able to look at two gene clusters within the same strain. And with both gene clusters, we had great success in improving yields significantly also. But what about discovery, right? Because we mentioned engineering, this compound still can be discovered even without engineering. So what about engineering? So the next story is something that we had done, and we were actually surprised when it worked, right? So this is Streptomyces rhodosporus. For those who know what dapamycin is, this is a strain that is commonly used and also very well known for making dapamycin. So the genome was sequenced in 2008. There are 29 predicted clusters based on anti-smash. And in and so we went back and we looked at the genome again. In the genome, there was, because this was sequenced in 2008, so there was some, there are misframes, there are iterated parts. The genome for the actinobacteria are highly GC rich, so high-seq alone was not sufficient. So basically now we have this mess up gene cluster or mess up biosynthetic pathway that we couldn't make head or tails of. But there was enough information within the genome to tell us that there was a regulator or activator within this gene cluster. And then our question was, could we activate the entire gene cluster that we have no idea what it makes based on the activator? And so we, again, using CRISPR, we did the knock-in. And if you see, this is actually our qPCR. Actually, this should be the band. This should be the levels in the wild type. And you can see, right, the genes are actually not detectable in wild type. And only upon upregulation of the R1 regulator, we see upregulations of all the other genes. So it's very nice. And again, upregulation based on new peaks, really, with the new upregulated protein, right? So with this, now we have a compound. And actually, it's a new molecule that we named ivermectin after the Sleeping Beauty, right? Because it only woke up after we activated it. But it was a highly potent anti-multi-resistant bacterial acting poly-in macrolactam. Okay, and this is the full gene cluster that was elucidated after the fact. And then with this elucidation, now we can actually pinpoint again from genes. We can pinpoint the different moiety of the molecule. And in terms of engineering, what we did after that was that we went in and at the different points. So each star is different sites that we had mutated, deleted, including complement, right? And if you see, starting from this molecule, we could make changes to the sugar groups. We can make changes onto the cycle, the macrolactam, and also includes demethylation, changing out the active site, the starter unit. So what this allows us to do is really very quickly, we were able to do engineering. We were able to engineer different analogs of the ivermectin. And then with this, we took the analogs, I think 1, 3, 4, 5, 6, 10 for activity assays. And what we found out was very interesting. Basically, just by removing the metal groups on this sugar, we are able to extend this into a broad spectrum antifungal now. Okay, by removing the metal group here, we actually lose activity. So the metal groups, all this, we actually lose activity in this role. However, if let's say we remove the OH and keep the metal group, we actually retain activity. So there was a lot of very interesting insights by being able to make the different analogs. And then really that will guide how we can engineer the organism even further. And this is really coming back to the story, right, where we want to be able to manipulate the genes or engineer the genes to make the different molecules, the different designer molecules, right? I promise you discovery, right? So with this, we went and we did look at Rhodosporus, Lividans, Penicillin, different strains across species, really looking at doing activation. So the question there was, could we activate the different strains? But a lot of times is based on genome mining. So we use genome mining to look for clusters of interest. Then we go in with different strategies to either upregulate based on open reading frame or activators, and then see if the regulation happened really. In this case, we were pretty successful. We could basically activate most of the strains that we were looking at. But then, and so, no, our first quest is done, right? But there's always a second quest, right? This is a secret quest, right? So the secret quest at this point was, so instead of one strain at a time, could we do this for a collection of strains? Let's say I have a collection, and there are various collections in the world. Within Singapore itself, we have the NPR collection that has about 50,000 or 60,000 actinobacteria strains already. So the question is, instead of one strain at a time, can we then scale this activation for a collection of strains? And in order to do that, I think there are some parameters we had to set up. So the question is, we need to have general approaches. If you remember the story about the Cas, why we need to have a Cas toolbox, because every strain behaves differently. So with this, we need to have a general approach that will allow us to tackle diverse actinobacterial. We need to have rapid approach. That means, again, I want to be something, it should be something similar to the Cas activation. It should be one step and it should be highly successful. I don't need to troubleshoot. I don't need to return. I don't need to multiple pass, right? So the question is, can we do that, right? For this, we actually turned to a different way of doing genetic editing. We went backwards, or we went back to something that was already known in the field. So this is the 5C31 integrase. The 5C31 integrase is a DNA recombinase. It's derived from Streptomyces phage 5C31. It can mediate recombination between the ATTP site and the ATTB site. And in current literature, as of I think last year, I saw it has been used in mammalian, plants, fungal, basically used across the spectrum, even across kingdoms. So it's very nice, and it seems to be very promiscuous. So as in, it can work across strains, and genus. So we also want to ask the question, how well will this work in our hands, right? So the first thing we need to do, and before any good genetic engineering is possible, we need to make sure that we could actually transform the DNA into the strain. So here, we wanted to, so using a empty, so a plasmid that has the Cas protein, but no guide RNA or no template, we compare the transformation of that into 23 strains compared to a strain that has the 5C31 integrase and the ATTP side, P side, but no other cargo, basic resistance. And here what we saw was very interesting. And where the Cas plasmid could only be transformed into 12 strains, whereas the 5C31 integrase can actually be transformed to 21 out of 23 strains. And so this is actually an overlap. Out of these 23 strains, about 10 strains can be transformed with either plasmid. 2 strains can only be transformed with the Cas plasmid, whereas 11 strains can only be transformed with integrase. I think so. This is a no-brainer, right? Of course, to be able to do across diverse actinobacteria, I'm gonna use the 5C31. And then with this, what is our strategy? And if you have heard throughout the talk, we were using activators, as specific gene cluster activators. We were also using enzymes within the primary metabolism that will help improve substrate levels, right? So here we use 5 different activators. They are either global acting activators or natural product or biosynthetic cluster specific activators. There are also here the fatty acid CoA synthase, which is acting on not as a regulator but also as a enzyme to basically improve CoA levels. Okay, so here we have our hopefully generalized protocol where we now will use the 5C31 to introduce 5 different cassettes where we will have a strong constitutive promoter onto all these genes. So okay, this is what we did. We took 54 strains that were derived from the Singapore collection. Basically, using one pass. That means we only did the experiment one time. We integrated 5 different plasmids into 54 strains. Whatever was successful, we took that. And so we had 124 unique combinations, about 400 mutants. And then we not only did genetic activation here, right? We also did cultivation activation. So if you remember, the story before was about genetic activation, but there is always a strategy, right, where we could activate the strain based on giving them more nutrients. And this is really the idea of OSMAD, whereas one strain makes many compounds, right? And really based on that, we have fermented the 124 combinations in 3 to 5 different medias. So with this, we then took the extract, we did LC-MS profiling, chemical profiling, and we also look at their bioactivity. So this would be antifungal, antibacterial, and also cytotox. Okay, so now we have very nice profiles, but we also have very systematic, comprehensive analysis. And really the question we really want to ask is, does it work, right? So we're doing so much really in hopes that we could at one shot be able to activate a very large library successfully, hopefully. So this is what happened. So each dot here, and this is basically a map of the the metabolites together. So similar scaffolds are clustered together, and each dot is a metabolite. And really, what we saw was that we had twofold increase in unique metabolites. That means we were making double the amount of metabolites compared to wild type. We were also making higher amounts of metabolite compared to wild types, but that data is not shown here. And then the next thing we wanted to ask, and that's something that people also ask, right? If you're making more, but are they the correct more metabolites, right? Because I could be increasing primary metabolites, I could be increasing something that's nonsense. And so that's what the bioactivity is. So in this bioactivity heat map, what we show here is bioactivity compared to wild type. So anything that lights up basically it means that it has more bioactivity compared to wild type. And here, looking at antifungal, antimicrobial, and cytotox, I would say, yes, we are making biorelevant metabolites, and we are improving the scale of that. So a lot of this could be not just activation of new compounds, but really scaling the previously observed compounds that were made in very low levels into much higher levels, so that we can see bioactivity and we can look at phenotype. So from this, we actually selected gram-negative activity, something that had really high gram-negative selectivity compared to wild type. We took this, we deconvolute, and we characterized. And this was a new tetra acid analog that has gram-negative activity. So this is the first example of a tetra acid scaffold with gram-negative activity. So again, very exciting and very easy to do. But we also came out from all this comprehensive profiling, right, with information on does it work? And I would say, I think it does. And we also have new insights on how to make it work more efficiently. So if you remember, we are doing 3 to 5 media fermentation, because we didn't know what should we do or how much should we push. And here, right, looking at, so this is the amount of media on the x-axis, and on the y-axis is how much compounds we have. So you basically need about 3 medias, and you reach about 80%. So any 4 medias will give you 90%, and then 5 media we consider 100%. So we don't need to go up to all 5 medias. A combination of 3 medias will be good. For regulators, this is regulators in 1 different media, regulators in 5 medias. This is the number of regulators in here. So if you add regulators up to 3 regulators, right, that would allow us to see complete benefit of what's required, right? And here we actually don't, so the question is there specific regulators that were better for others? And because we're doing on such a diverse spectrum of actinobacteria, in this case, any 3 regulators seems to work pretty well. So it's not even specific regulators. So I think in the end, I think from this experiment, really comprehensive experiment, to go for the question, can we do high throughput activation on the strain library? And I think the answer is yes, right? So we have new protocols where we have integration. We show that integration of 5 activators. It could be biosynthetic specific activators, it could be global activators, and it could be primary metabolite enzymes. Any 5 or any 3 from here, and really looking at also 3 different media combination, we are able to double our chemical space, but we also enable discovery based on improved bioactivity. So we are also doubling the biorelevant molecules. And then really coming back, right, to our initial question was that can we engineer molecules? And if you were to take a step back, really, I would say with the tools that we have, really starting from the CRISPR story, then looking at the integrase, and also looking at the cultivation optimization, I would say that we have tools that will allow us to go from genes to chemicals, and where we can now even supercharge the cells to make molecules that were not observed before. And really that will be what we are very interested to do. And I say, yes, that's something that we can do now instead of something that we hope we can do, right? So then, this is the adventure, and hopefully the start of many other adventures for our streptomyces discovery and engineering work. But then going back to the central dogma, another story before, after molecules, right? We need to go back and say, thinking about all this, the actinobacteria was also very, if it makes a lot of molecules, it also means that it has a lot of chemistry diversity because it has a lot of proteins. It has a lot of diverse biocatalysts. So here we are interested to see genes to proteins. Is there any proteins in the actinobacteria that we could exploit too? So one example of this exploitation is actually not exploitation, it's sustainability, was the example of PET. So you have PET bottles. Those actually can be recycled now based on using chemical and mechanical processes. But repeating chemical, mechanical recycling can degrade the polymer integrity so that the mechanical performance over time will actually reduce. So you can only recycle it up to 6 times. But an alternative is to then depolymerize or degrade the PET into its monomer, that is TPA and EG, and then repolymerize to PET. So that will ensure that, you know, we break down completely to monomers and we do a chemical polymerization back to recycled. So the recycled PET at this point will have very high structure. It will not be like the ones that will go through chemical and mechanical processes. So in 2020, there was a very nice paper published where they used a LCC, it's a leaf composed cutanase, that will allow us to break down PET to TPA and NG. And this is all enzyme based, right? So it's enzyme that is used at higher temperatures but able to break down to monomer. And interesting enough, so although it's called a leaf composed cutanase because it was found in metagenomics of a leaf compose, it was actually proposed that this enzyme is actually actinobacterial in nature. Then this started off with the question, can we find equivalent enzymes in our library, right? So this is a sequence celebrity network where this is where the LCC is. Within our collection, our own collection, we also picked out a few enzymes to test, and we call it the Cyber 1, 2, and 3, and 4. And this is where peptase, where another enzyme that can break down PET is also found. So within this, we took the sequence, we synthesized the sequence. So in this case, we had tools like DNA synthesis, cloning, expression, and assays to help us. So for enzyme discovery, what we did was here, we generated from basically different constructs, looking at different texts or different versions or expressions of one gene. So each row is basically about, I think, 10 to 15 of one gene. And out of this, we tested for expression. So if you look, although all genes created equal, but folding is not equal. So here we have, for example, the Cyber 1 prefers to be his tag protein, Cyber 2 too, but Cyber 3 uses this tag 11, is a 11 amino acid tag called anti-11. And then the sumo is another protein preferred by another protein. So really different proteins fold differently, and there are different aids to help the protein folding. So with this, we selected the best of each gene. So in this case, it would be this, and really outlined our box. And then with this, we test the polymerase, depolymerase activity or capability of the gene. So this would be under 25 degrees, under 40 degrees, and under 50 degrees, looking at TPA and MHET production from PET. So if you remember correctly, the PET breaks down to BHET and then is broken down to MHET and TPA. So looking at this, we have actually, what's interesting is that the Cyber 1, so this 171 construct is actually stable over different temperatures, and we still get TPA production. And then with a bit more engineering, we also have another, I think it's a mutation of 822. So this is called Cyber 822 from 171, derived from 171, that is able to basically withstand 1 week at 50 degrees, really breaking down the PET to TPA and also MHT. And interesting enough, this enzyme interest is actually 60% similarity, similar to LCC. So it's actually not very similar, but it's able to still do its job. And then I think with this is another adventure for us looking at protein engineering, protein discovery, and also for the actinobacteria. I hope I also convince you guys on its possibility, either chemistry or enzymatic. And then also with this, I'll end our story. But a lot of this cannot be accomplished without our collaborators. So this will be collaborators over across A-Star, also across the ecosystem, for example, in UIUC and with Mingzhu over at Taiwan right now. So, and then with this, I would like to thank the audience and also